Hello, everyone. Today I will be giving a speech on the topic of gene editing, and I will be talking about the advantages of gene editing, the history of gene editing, and also the disadvantages of gene editing. I would like to bring your attention onto the case of 1-year-old Layla Richards. She is a little girl who was brought into this world with leukemia from a very young age, at birth, diagnosed instantly. Doctors and her family tried everything they could, treatments, types of therapy, none of them worked. That was until immunologist Wasim Kassim from the Great Ormond Street Hospital for Children in London reached out and suggested the use of gene editing therapy to hopefully fix or at least alleviate temporarily Layla's condition. And after an extensive surgery and the extraction of T cells and using enzymes to put in good genes and take out faulty ones in Layla, her life was saved and she was in remission now and now able to live a normal life. Today, my topic is on gene editing, like I said, and I'm also here to discuss with you some of the conditions someone you may know, or the average person may have that may be treated or somehow eliminated in the future through gene editing. I am qualified to discuss this topic with you because, one, I'm a STEM student, and because of the research I have completed for this presentation. A little preview. I have 3 main points and in my body speech, and those 3 will be, like I said, the history of gene editing, the advantages of gene editing, and the disadvantages or adverse effects that gene editing could cause. There we go. Now, let's begin with my background on my topic. My first main point is on the history of gene editing. In the 2017 paper, A History of Genome Editing in Mammals by Alamunda Fernandez, she states that the exchange of indigenous genomic sequences has been known for decades, which is true because it was pioneered, gene editing was, by Dr. Oliver Smithies. He discovered that DNA could be placed and taken out of chromosomes in mammals as early as the mid-1980s. Their research helped understand stem cells, which you can grow into basically any cell you need. It's a very diverse and amazing thing our human body does. Him and his colleagues, they are the ones that paved the way for modern gene editing that we know today, also known as CRISPR gene editing. Then, in the 2018 paper, The History of CRISPR-Cas from Encounter with a Mysterious Repeated Sequence to Genome Editing Technology by Mart Karpovich, he states that the CRISPR-Cas system was finally proven in 2007, which is when CRISPR was invented. CRISPR allows for a much more precise gene editing process than previous technologies were able to that Dr. Smithies and them worked with. Contrary to popular belief, actually, gene editing and CRISPR uses natural proteins to do the whole process. So there's no artificial genes going in and out. It's just how you shape them is in an artificial way rather than naturally with nature letting that process happen. In 2017, actually, researchers in China, they treated a man with lung cancer with CRISPR, and it was successful. Now that you're familiar with gene editing history, I will now move on to my next area of discussion, which is the advantages of gene editing. In the journal titled CRISPR-Cas9, a new and promising player in gene therapy, Liu Zhaoji concludes that CRISPR-Cas9 technology can be applied for therapeutic purposes for several ways. And those ways being, one, it's very cost effective and it's very easy to use for the average person. Average person being the scientists that work on it, average. So also the ability to change genetic code from the genome sequence very easily, and also to flush diseases such as like HIV or cancer from a patient effectively. Furthermore, in a 2021 article, CRISPR-Cas13 system as a promising and versatile tool for cancer diagnosis, therapy, and research by Farikin Pelez suggests that the CRISPR system can actually be used for treating cancer patients effectively, as well as diagnosing the patients and also furthering cancer research and research with just the CRISPR system and its functions as a whole. My third main point are some of the cons and disadvantages of gene editing. One of the major disadvantages is the lack of regulation on CRISPR technology, which Clovis Tanning in the 2017 journal CRISPR and Cas9 in Insects, Applications, Best Practices, and Biosafety Concerns, shares that the current international and national regulations do not provide adequate guidance and oversight. This is a trend in the gene editing industry that can be found everywhere, no matter what they're using the gene editing for, whether it be seeds or animals or pharmaceutical use in humans. It's an issue that everyone, including lawmakers and activists, point out because the lack of regulation in the industry, as can be seen in other types of industries, can lead to an abuse of the technology. And lastly, the journal CRISPR Ethics Moral Considerations for Applications of a Powerful Tool in 2019 by Carolyn Borkowski. She insists that there is some ethical issues that the CRISPR tech could pose, such as a very blurry distinction between therapeutic and enhancement applications, leading to potential subtle or obvious exacerbation of inequalities, or the possibility of the tech to be used for eugenics, such as a term that's bounced around as designer babies, or I want a kid with blue eyes and blonde hair, let's say, for example. That in theory is possible with gene editing tools. In conclusion, I have discussed the topic of gene editing today, and there's three main points: the history of gene editing, the advantages of gene editing, and the disadvantages of gene editing. And before I leave you, I would like to leave you with this statement. As gene editing technology naturally evolves and progresses into a more normal application into modern medicine practice, I ask that you not welcome the technology blindly with open arms, but to keep an open mind about the technology and the possibilities of gene editing so you're able to make an informed decision on the technology without improper information beforehand. Thank you.